Okay, so let's go ahead and get into the third part of the lecture on the molecular biology of the gene. So we talked about what DNA is, the history of DNA, the basic structure, that double helix shape we've talked about, the bases associated with it, how the base pairs A to T, C to G. And in the last lecture, we talked about how DNA replicates, because during the S phase, that replication has to happen in order to make more DNA. So put this in context with the cell cycle. The cell rips itself in half during the cell cycle. One cell becomes two, and they should be identical to each other. Well, to be identical, you better have identical DNA, and then you rip the DNA in half. Same DNA goes into each of the new cells, and then both of those cells have to come back and increase the volume of their DNA. And then they rip in half again, and this continues over and over and over. And remember, roughly 50 times before the cell eventually is worn out and the cell dies. So when we look at the big picture of DNA, it's just crazy to think we have about 100 trillion cells that make up the human body. I mean, this is an average. Everybody's a little bit different. But 100 trillion cells. If I could take all of your cells and suck out all the DNA, there's about 6 feet of DNA per cell. So if I could suck it out, I got about 600 trillion feet of DNA. That means literally I could line it up and go from the sun and back to the earth about 68 times. I mean, that's a huge, huge amount of DNA. So anytime we have a single strand of DNA, we can figure out what your genetics are. So we talk about crime scene analysis. Oh, the perpetrator, the criminal, the whatever you want to call them, left a cigarette butt at the crime scene. Well, they were smoking. It was in their mouth. There's saliva on it. There's cells in our saliva. Those cells contain our DNA. That's all it takes to place somebody at a crime scene. Doesn't convict them, but it's just a piece of evidence to support the law enforcement officials, or to allow somebody to go free who is not guilty based on DNA evidence. So roughly every second, about 50 million cells are finishing up the cell cycle, finishing up that 50th time, and they die. Or they're getting pushed, their self-destruction button is getting pushed, and they're getting destroyed because something's wrong, they're messed up, there's not enough energy, whatever the case may be. So 50 million cells. So if you are at a point in life where your body is just trying to maintain, just replace the cells that die and maintain your body stature, we need to build about 62,000 miles of DNA every second. I mean, that's just an amazing amount of replication that's happening. And the goal is it all replicates correctly. So this is why we need energy. We have to eat. We have to bring in calories to just make more DNA, to make more cells. We need a massive amount of calories just for that basic function. So go back to the food chapter when we talked about which types of food should you eat. Now the good news is anytime you eat anything, I don't care what you had for dinner, chicken sandwich, I had some toast, I had a bowl of lettuce, a tomato, every single one of those things you've eaten contains DNA. Every cell has DNA in it. So you're eating something that was living. It has DNA. So we're using that DNA to make more for ourselves. All right, so now what we want to explore is how does our DNA turn into blonde hair, blue eyes, and those physical traits? Well, this is done through the process of gene expression. So we're expressing a gene. We're expressing a physical trait. We're going to express our phenotype. We're using the genetics or the genotype to actually show a particular characteristic. So DNA acts as the menu. That is the beginning point. Let's keep that in mind. DNA is our menu. So whatever code you have here serves as the template. DNA gets turned into RNA. RNA is kind of this shorthand code, this intermediate, that then gets turned into amino acids, a string of amino acids. And the amino acids express the physical trait. So this process, transcription, translation, that is how we take DNA and turn it into brown hair, blue eyes, etc. Now, what we're learning is it is very possible to go in and maybe I want to shut off a trait. Turn it off. Let me block it. So now we block this process and I don't express my trait. Or do we want to turn it on? And it's a trait that we carry but we don't normally express. Do I want to flip the switch and now express that particular physical trait? That's all part of biotechnology. Is this a good thing? Is it a bad thing? Everybody should form their own opinion on this and support their opinion based on information, facts, and evidence. So the key here is, we've already talked about DNA, but the next key is this stuff called RNA. What is it? What is RNA and how does it work? So RNA is known as ribonucleic acid. DNA was deoxyribonucleic acid. RNA is ribonucleic acid. RNA is made up of 3 components. Start with the sugar backbone. The sugar here is ribose, so a little sugar house. In DNA we had the deoxyribose. So there's our ribose, our sugar house. Our second component is going to be a phosphate group, just like in DNA. So here's our phosphate, little phosphate chimney. And then our third component is going to be a nitrogenous base. Okay? Just like in DNA. But, here's your big but. RNA uses a base called uracil instead of thymine. So it will have cytosine, check. It will have adenine, check. It will have guanine, check. But it does not have thymine. Instead, it uses a base called uracil. Okay, that's one significant and very important difference between DNA and RNA. The sugar, our RNA is using ribose versus deoxyribose. That's another important difference. And then the third important difference is the fact that RNA is just a single strand of information. It is not the double helix. It's this straight single strand here. So you got 3 critical differences between RNA and DNA. It makes a big difference when we start talking about how do we create a protein and express a trait. Okay? So you get this big string of RNA letters. In this example, C, A, U, G, and then whatever. Maybe it's A, 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 U, G, C, C, G, etc. That's all based on the DNA. But that RNA information is what will become amino acids, or it codes for amino acids. All right, so RNA is read in these 3 base combinations called a codon. Okay, the codon is 3 letters long, 3 RNA bases. So 3 base combination equals a codon. So UA or UUA equals a codon. And that codon then is translated into an amino acid. All right, so each codon is represented by one of 20 different amino acids. So any 3-letter codon you can create from the DNA alphabet, or I'm sorry, the RNA alphabet, and your RNA alphabet again is A, U, C, G. Those are your 3 letters to work with. Create a 3-letter word that will be represented by a specific amino acid. So GAG, there's a codon. GAG, if you come over to the chart, find GAG, and GAG is down here. That represents the amino acid glutamate, or codes for it. So let's go with something simple. AAA. If you had a codon AAA, well, that codon is going to code for or represent the amino acid lysine, etc., etc., etc. Any three-letter combination you can create with RNA is going to be found on this chart in front of you. That's how we would then take that RNA and turn it into a specific amino acid. Or, more importantly, the ribosome is going to do this. That's our protein factory, the ribosome. So remember, back to cell structure. Ribosomes build proteins. They sit on the endoplasmic reticulum, the little bumps, so we call them rough ER, and that is going to be where our proteins are built. So DNA acts as a template to create this stuff called messenger RNA, or it's abbreviated little m RNA. So DNA turns into mRNA. The DNA is transcribed, so that is a key word to remember. Transcribed into the messenger RNA strand, and this is going to happen within the nucleus of the cell. So try to fit a cell in here. So there's our cell, here's our nucleus. Inside of it you have, there's your DNA strand. That gets turned into this little thing called messenger RNA. There it is, inside the nucleus. Now, something important happens here when we create messenger RNA, and we create this string of information. It gets modified a little bit. It gets kind of organized and cleaned up a little bit before it actually leaves the nucleus. So what will happen is certain pieces of the RNA strand are cut out of it. These are known as introns. So introns are removed from the RNA strand, and they are left in the nucleus. Right now, scientists honestly don't know what they do. They don't know what is the function of the intron. It could be really critical. We just don't understand what it does yet. It's still being learned. So you take out these introns, these sections of the RNA strand, and you're left with these things called exons. Now, exons are the remaining chunks of the RNA strand. They are spliced together. They form what we call the mature RNA strand, and then that physically leaves the nucleus. So introns stay in. So those stay inside the nucleus. Exons get spliced together. So splice these together here. And then it becomes the mature, called the clean, the perfect RNA strand. That is the piece of information that leaves the nucleus and is making its way over to the ribosome. So this is all in here. This is all part of transcription that's happening inside the nucleus. That's supposed to be an A. Transcription. Okay, sorry about the sloppy writing, but that's supposed to say transcription inside the nucleus of the cell. Now, once it is done with transcription, it's going to exit the cell, it's going to head over to a ribosome, and it's going to be translated. So that code of mature messenger RNA, all those little letters strung together, will go to the ribosome, and it will be translated 3 letters at a time into specific amino acids. So we'll take a look at that in our next lecture. How does messenger RNA, the mature stuff, turn into proteins?